Greetings. Today, I'll be giving a brief summary on the paper, A Small Molecule Kinase Interaction Map for Clinical Kinase Inhibitors by David Lockhart et al., which was published in Nature Biotech in 2005. This paper describes an experimental approach to assess the specificity of kinase inhibitors that directly and quantitatively measures the binding to the ATP site. The key assay components are human kinases, which are expressed as fusions to T7 bacteriophage, and a small set of immobilized probe ligand that bind to the ATP site of one or more kinases. In figure 1a, for the assay, tagged kinases and immobilized beta ligands were combined with the free test compound. If the free test compound binds the kinase directly or indirectly, it occludes the ADP site, and hence fewer protein molecules can actually bind to the immobilized ligand which is on the solid support. But if the free test molecules or compounds do not bind to the kinase, then tagged proteins are able to bind. Quantifying the amount of fusion protein that are bound to the solid support using either the traditional phage assay or qPCR gives the result. They initially built an assay for P38 MAP kinase and chose SB202190 as an immobilized ligand. BERB and VX are two other inhibitors that target P38 alpha. Figure 1D shows that the competition with unmodified SB202190, SB203580, BERB, and VX decreased by 1,000-fold or more the amount of tagged P38 bound to the solid support, whereas neither 202474 or parvalinol A had an effect. As BERB binds predominantly in a position adjacent to the ADP site, it affects conformation of the ADP site indirectly, whereas the remaining three bind directly to the ADP site. Thus, they infer that this assay can detect allosteric as well as direct binding interactions. Figure 1c shows how they determine the affinity of the interactions by quantifying the amount of tagged P38 bound to the solid support as a function of free test compound. To avoid the need to find a unique ligand for each and every kinase, they sought molecules that bind with high affinity to the ATP site of multiple kinases. One such molecule was staurosporin, and they tested out 113 distinct protein kinases with staurosporin and validated that there is very high correlation between inhibitor binding affinity measured in competition assays and inhibition of enzymatic activity measured in traditional enzyme or cell-based assay. To organize the result, they performed this 2D hierarchical cluster analysis of the specificities of the inhibitors to the 113 kinases. The lighter color corresponds to tighter interactions. The authors inferred that the binding assays have several advantages with respect to the traditional in vitro enzyme activity assays because it's faster. The entire panel can be run in a single experiment in parallel using similar reagents. It requires low amount of kinases. It can test a wide dynamic range of binding affinities, and it can compare directly. And also, it circumvents the need for conventional protein production. Thank you.